Thanks everybody for coming here. It's great to see everybody, new, old friends here at the DVOC meeting to hear about bird sense of smell. And one thing I like to do when presenting to bird club, ornithological clubs, is ask uh, you guys some questions. Does anybody have any stories about what you heard about the sense of smell in birds, or what you might know about the sense of smell in birds, or any observations? Anybody? Yeah.
If I remember right, Audubon did a study with black vultures and turkey vultures on scent
Yeah.
and smell.
Yeah, I'm going to mention that.
Studying it going back, that'd be the mid 1830 maybe he was doing that.
Yeah, I got the year on the slide, but I can't put it off the top of my head. Yeah.
But going out on pelagic bird ships, lots of seabirds,
Yeah.
a very strong sense of smell.
Yeah. You throw out the chum, yeah, bucket, and they come right in. Yeah. Yeah. I went on sorry, someone's gonna say something? I went on a pelagic trip with my PI, my boss, who loves the sense of smell, but he's not really into birds. He loves the sense of smell, and the only picture he took the whole time on the pelagic trip was of the oil bucket. That was the only thing he cared about. So everybody else was getting on the storm petrels and all that. All right, so yeah, so birds. What do we know about them? Well, there's a message. Oh, da, 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 da. I know that in the past it was believed for a long time that birds couldn't smell. Right. So I will get into that as well. So yeah, so birds, they got every color in the rainbow here. They're super colorful. Sometimes they have these elaborate ornaments, like on that spatral tail on top right, King of Saxony, bird of paradise, top left. They can have crazy displays for sexual displays. And so it's very obvious that these organisms' vision is really important to birds. Vision is super important. But there's a darker side to these bright colors, and that is that in ornithology, there's been this prevailing thought, which was just described in the Zoom chat, that birds are mostly diurnal, they're often colorful, so therefore they must rely on vision at the expense of olfaction. And this idea was really synthesized in this 1905 paper from Alex Hill in Nature, where he used morphological and behavioral evidence in the turkey to conclude that olfaction is not really very relevant to bird behavior and ecology. And his results were bolstered by earlier experiments from some of the most famous biologists of all time, John James Audubon, 1826, I guess was the exact year, where he buried deer carcasses underground, just talking about, and he found that turkey vultures were unable to navigate to those carcasses, so he concluded the turkey vulture could not smell. And then there's a part of Voyage of the Beagle, if you read it, where Charles Darwin is in Chile, and he's in front of this row of these Andean condors. So this giant, huge. I mean, you guys know one of the biggest flying birds, Andean condors, and they're all chained up. And he's holding a piece of meat in his hand that's wrapped up in paper, and he's showing them the paper, and they're all just kind of uh, standing there, just looking at him, not doing anything, like just there, just like all nonchalant. And then he rips the paper and exposes the meat, and they all start flapping around wildly. And he concludes, "You couldn't have fooled a dog in this way." So, for a long time, this was the thought. And then it wasn't until you have to fast forward to the 1960s that we had the first ever behavioral experiment showing the sense of smell in birds. So what happens here, I kind of want to point, but I'm too far away from the screen. Maybe my mouse. Then I'm looking at the screen, which I don't like.
Put up the camera lower left.
Uh, I got this. Oh, wait, can you see? Yeah, yeah. Okay. So in this experiment, the pigeon was put in this chamber, and it steps up on this perch, which is called the hopper in the figure, and then it puts its beak through the keyhole, and when it does that, it gets a seat. And so quickly, the researchers were able to train the pigeons on this, and then once the pigeons learned that, they started flowing amyl acetate through the chamber. And this is an odor that smells a little bit like bananas. And when the pigeon now goes in the hopper and pecks the keyhole with the amyl acetate flowing, it doesn't get a seed anymore, it gets a shock. And so the pigeons quickly learned to only go on the hopper perch and peck the keyhole when the amyl acetate was not flowing, when the control air was flowing. And this was published in Science in 1966 from Florida State. And this was the first behavioral experiment showing sense of smell in birds. In the past 20 years, there's been a lot more work, sort of an explosion in the last 20 years of bird smell research. So Julie Hagelin at Swarthmore University, maybe a lot of you guys in this room know this. If you pick up a crested auklet and sniff it, it smells like tangerines. This crested auklet seabird in Alaska smells like tangerines.
You're allowed to just pick them up.
So I highly recommend when you go on a birding trip to Alaska. You see a crested auklet, you just take license to grab it and sniff it. So then she put the crested auklets in this Y maze where one side had fresh crested auklet feathers, as it says there, and one side was a control. And then she found that the crested auklet preferred to be on the side with the crushed feather odor as opposed to the control odor. And then when she took the feather odors and determined which odors were in the feathers and took just two of those odors and placed those odors on one side of the Y maze, so the one side of the maze, the auklet preferred to be on that side. And when she used the amyl acetate odor, like in the pigeon, as a control, there was no preference. And with a mammalian odor, there was an avoidance compared to the control. And then, I have about 4 or 5 examples here that I'll just quickly go through. So for individual recognition, this was interesting. The researchers here from Europe, they had a chamber with 2 sides here, and there's a fan that blows in the odor to each side. And on one side is the female zebrafinch's, female zebrafinch's own egg, and on the other side is a conspecific egg, and so not the zebrafinch's own egg. And after 3 days post-laying, there was no preference for either side of the chamber. But after 10 days post-laying, the females preferred their own egg as opposed to the conspecific egg. And the authors took this that the zebrafinches could smell their own egg versus another egg. And then for nest material selection, these researchers put out nest material for starlings that were either had a vanilla scent added to them or a control with nothing added, and found that the starlings preferred to pick the vanilla odor nest material. And they also did this with this milfoil, which is kind of like a seaweed-like scent, and found a preference there too. So potential olfactory roles for nest material selection. And then Gabby Nevitt's life work, she's at UC Davis, has really been shown to show the sense of smell in procellariforms in the seabirds. And so what the researchers did here is in the top left, they put out the fish oil here in these dots. And in the bottom right, we're looking at a map of the ocean here. In the bottom right was the starting point of radio-tagged albatrosses. And they found that as long as the albatrosses start downwind of the fish oil, they're able to navigate many kilometers across the open ocean to the location of the fish oil in a flight direction that is supportive of the use of olfaction. I think this is the last example. So white storks from Europe, this is a more recent paper, someone made an observation that storks kept appearing in freshly mowed lawns. Someone was noticing this. And so the researchers took chemicals from freshly cut grass and just put them in a field that was not freshly cut. And so here, this blue line is the direction of the wind. And these 2 blue dots are just locations where they just put water on the field, so like a control. And then this green dot, this green dot is where they put the leaf alcohol, the odor associated with cut grass. And they found that radio-tagged storks, which are at the starting point of each one of these arrows, were able to navigate to the leaf alcohol spray, even though it wasn't freshly cut, just a spray, as long as their starting location was downwind of the spray. So these yellow lines are storks that went to the green spray, and these dashed lines are storks that just didn't move. So I don't know why they made them into lines, but they just stay where they were. And then finally, there's a few lineages of birds that have for a long time known to be able to smell. So kiwis have an excellent sense of smell. You might know this. They're the only bird that has their nares, nares or nostrils in mammals, but nares in birds at the tip of their beak as opposed to the base. So they breathe through their whole maxilla when they inhale. And of course, they're foraging at night for invertebrates under the soil. So they use sense of smell. And then indicator honey guides. There are some, these old papers are amazing about honey guides so honey guides eat wax, their diet consists of wax, and they use sense of smell to locate the beehives. And so in these papers, they put candles in the tree and the honey guide wouldn't come to it. And then they lit the candle and the honey guides came in and started drinking the candles. So very cool experiment there. And then vultures, turkey vulture, as it turns out, as we know now, has an excellent sense of smell. It's thought that the deer carcasses that Audubon was using were too rotten, or that he was actually looking at black vultures. So I'm not sure if it would stand up to peer review today. But that's great at the behavioral level, but what about at the molecular? So at the molecular level, this is how smell works. We inhale the air through our nostrils, and then the air passes along this tissue at the top of our nasal cavity called the olfactory epithelium. And on the surface of the olfactory epithelium are these olfactory sensory neuron cells. And on the cilia projecting out from the olfactory sensory neurons are the olfactory receptors. And the olfactory receptors are the front line. They're the interface between biology and the environment, where the odor comes in from the environment into the olfactory receptor, which are 7 transmembrane G protein coupled receptors that sit on the plasma membrane of those olfactory sensory neurons. And the odor comes into the olfactory receptor, and there's a conformational change in the olfactory receptor. And this initiates a signal cascade that allows us to then eventually perceive and respond to the odor. And there are so many different kinds of odors in nature. They can be big, small, have a ring, doesn't have a ring, has a sulfur, doesn't have a sulfur, positive charge, negative charge. Even these two enantiomers, which are the same chemical formula but a different structure, can have different odors. And so it's estimated that with all these odors, humans can discriminate, as it says there, more than 1 trillion different olfactory stimuli. And the way this works is in this combinatorial fashion, where this is just a cartoon, but say in this cartoon organism has 25 olfactory receptors in this 5x5 grid. And this odor in this example comes in and activates this olfactory receptor. But then this odor comes in and it activates 2 olfactory receptors. So odors can activate multiple olfactory receptors. And then this odor comes in and it activates these 3 olfactory receptors, but it activates the olfactory receptor in the second row all the way to the right, which is the same olfactory receptor as the odor right above it. So an olfactory receptor can also respond to multiple odors. And then this odor here comes in and activates a whole suite, a whole combination of olfactory receptors. Sorry, this odor comes in and activates all these olfactory receptors. And then this odor can come in and activate some olfactory receptors, but then deactivate or antagonize other olfactory receptors. And then if you take these same 2 odors and put them in a blend together, in a context together, they can activate and deactivate a whole new combination of olfactory receptors that they didn't when they were by themselves. So this is how you get to 1 trillion different odors that we can perceive. And so because there's so many different odors, now, olfactory receptors are the largest gene family in all of vertebrates, with over 1,000 olfactory receptors in many species. So on the graph on the left, on the x-axis, we have the number of olfactory turbinates. This is a morphological structure in the nose of these mammals. And on the that was the y-axis. On the x-axis, we have the number of olfactory receptor genes. Now you can see that many mammals have multiple thousands of olfactory receptor genes. And if we just filter, this includes pseudogenes, this graph, but if we just filter for functional counts, we see that many mammal species have over 1,000 olfactory receptors, with elephant at the top as the champion mammal with 1,765, and humans down there with 372. So that's great about mammals, but we're all here because we love birds. So do birds have a lot of olfactory receptors? So to answer this question, we had to dive into the bird genomes. And so what we do is we build a known query file of previously known olfactory receptor sequences. So we take olfactory receptor sequences that are already known and basically do a Google search, just like a Google search, against the chicken genome and find the matches, find the hits. And so when we get the hits, we then filter them for like open reading frames, have an intact transmembrane domains, length, no overlap, etc. And then we take the filtered hits, align them, and build a tree. And when we build the tree, it almost always forms this stereotypical shape. So each tip here is showing an olfactory receptor in the chicken. And this shape shows the three groups of olfactory receptors in birds, which down here in the green are the class 1 or alpha olfactory receptors. They detect water-soluble odors. Right above it in the yellow are the gamma or class 2 olfactory receptors. They detect airborne odors. And above it in blue here are the gamma C olfactory receptors. And these gamma C olfactory receptors are very special. They're the vast majority of olfactory receptors in birds. They have these very short branch lengths, so very high sequence similarity, and they are only found in birds. These olfactory receptors are not found in any other organisms, not even crocodiles, the closest living relative of birds. So we looked at this in the chicken and we found 473 olfactory receptors in the chicken. So even just the chicken has over 100 more olfactory receptors than human. And the reason why we did the chicken first is because the chicken has this excellent genomic resource. This is a so-called T2T, telomere to telomere, gapless genome. And it's a very, very high quality, the highest quality bird genome. And the reason why this is important is because there's actually a second myth associated with bird smell. And that is that short read or Sanger sequence greatly undercounts the number of olfactory receptors. So I'm going to explain this. So basically, when people were sequencing genomes of diverse species, this really started in earnest in the 2010s. And back in the 2010s, the main technology available was Sanger sequencing or short read Illumina sequencing. And so people were sequencing bird species, finding out how many olfactory receptors they had, and publishing it. And when they did this, they confirmed or supported things that Audubon, Alex Hill, and Darwin reported on, that birds have way fewer olfactory receptors than mammals. So you can see here in the Illumina assemblies here, so there's 5 species here, and we have an Illumina, or short read, or Sanger assembly, and a long read assembly. And you can see that the short read and Sanger undercount the number of olfactory receptors compared to the long read. The long reads only became available in a diverse species in the 2020s, maybe in about the past 4 or 5, maybe 6 years. And so for a decade, we thought birds had way fewer olfactory receptors than they actually have. And when we look at the olfactory receptors that are being undercounted, they're almost all gamma C olfactory receptors. So these blue numbers are showing the number of gamma C olfactory receptors recovered from the different genomes. And we can see that in the Illumina especially, only 2 were found, for example, in the Anis hummingbird there, compared to 86 in the long read. So why are they undercounting these olfactory receptors? Well, the reason is the gamma C olfactory receptors. I'm going to explain this in a minute. I know this is not nice to look at, but the gamma C olfactory receptors are not located throughout the chicken genome. They're not here on this chromosome and that chromosome and over here. They're all in these giant clusters. And in the chicken, there's just 3 giant clusters. And although this says chromosome 33, this is actually looking at the giant cluster in chromosome 29. And so, for whatever reason, and each one of these little lines here is a different gamma C receptor, and you can see them all spaced out in this cluster right next to each other. And flanking these gamma C olfactory receptors are down here are a lot of repeat elements. So these repeat elements are just the same set of nucleotides over and over again, AG, 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 whatever, or in triplicates or whatever it may be. And so these areas of the genome are highly redundant as you go across them. And so what happens is this is how the sequencing technology works. I'm not exactly sure why we did the dogs, but we did the dogs. So if you have a long read genome, you're assembling the genome with long reads. You're assembling the genome with big pieces. So if you have four dogs and you have big pieces of the dog, like here, you can see that there are four dogs. But if you're assembling the genomes with very small pieces, like in short reads, really small pieces, and these pieces are all very similar, you might conclude that there's only two dogs, a white dog and a tan dog. You can't distinguish between these dogs. These small pieces are too highly similar. So this is why the short read technology was undercounting these olfactory receptors for so long. But nowadays it's 2026. So we have long read genomes from all across the bird phylogeny. So there's over 200 available. We've looked at the olfactory receptor repertoire in almost 200 species. And we found about 50,000 olfactory receptors total for an average of 258 per species. The range varies widely, and I'll get to that in a moment. And about 80% of these are that gamma C type of olfactory receptor. And so here's two examples of what it looks like. On the left, we have kittiwake, so the oceanic gull, and we have the alpha, gamma, and gamma C. And on the right, oh, this used to be a cuckoo. I used to have a cuckoo here, but now it's an ant shrike. All right, alpha, gamma, and gamma C. So this is what it looks like when we put just one species olfactory receptors on the phylogeny, but what happens if we put multiple species on the same phylogeny? Well, it winds up looking like this. And so here we have 6 species: swift hummingbird. This is a wida, strixoleuco, tawny owl, northern gannet, and mute swan. And here's the alpha, gamma, and the gamma C. And the alpha and gamma in black are grouping as you would expect them to. So your hemoglobin gene is way more similar in sequence to a chimpanzee's hemoglobin gene than your hemoglobin gene is to your insulin gene. That makes sense? So we would expect genes from different species to group together. So and that's how it looks for the alpha and gamma. So for example, here on this tip in the gamma, we have one gamma receptor from swan, gannet, owl, songbird, hummingbird, swift. They all group together. They all have that olfactory receptor. But that is not what the gamma C look like. The gamma C, which are shown in colors, form these species-specific clades, where all gamma C olfactory receptors from one species are more closely related to each other than any gamma C from another species. And so this is a really strange, really striking, unusual pattern, and we wanted to look into it more. So how does this pattern arise? Well, if you look at species with low levels of divergence, for example, there's only 9 million years separation between these two old world warblers, the gamma C are not separated, as you can see there. And then if you could look at 16 million years separation, we were just talking about swans, between this duck and these swans, you see there's a little bit of structure in the gamma C olfactory receptors, but it's still mixing. But by 25 million years of separation, like between these two suboceans, this mannequin and this tyrant flycatcher, there's separation between the gamma C. So how does this happen? So one idea is that it's just gene duplication. So this pattern could be explained by each one of these species starting out with just one gamma C olfactory receptor, and then they all experience duplication. That would explain this pattern. But that's not very parsimonious. It's pretty unlikely that every lineage of bird would just experience this duplication. So we're not going to go with that. So there's a term for this pattern called concerted evolution. And concerted evolution can operate in several different ways. So if you think about, if you look at a chromosome here and you look at genes that are in tandem with each other in a cluster, for example, the gamma C. And here we have a mutation or a substitution in one of the gamma C genes, which turned it from blue to red. And during the generations in meiosis, during unequal crossing over, the chromosomes have these clusters, and they're unable to line up exactly correctly because they're confused. I know chromosomes aren't confused, but because the sequence is so repetitive, they line up incorrectly. And so as they line up incorrectly, generation after generation, you go from all blue to all red. So this would also explain the pattern. But when we look more carefully at the nucleotide sequence, that's not what we see. So if you look at this web logo, this image shows an alignment of all 419 chicken gamma C olfactory receptors, and we're looking at the nucleotide identity at each position in the alignment. And if the nucleotide is tall, like here in these green boxes, that means that there's a high level of similarity between the olfactory receptors. If the nucleotides are short, that means there's a lot of diversity. For example, at this position here, there's T and C there, so there's diversity. But we find these really surprising huge blocks, which are in the green squares, of near complete nucleotide sequence homogenization, where all 419 chicken gamma C olfactory receptors have the exact same sequence. But they're interspersed with regions of high diversity, for example, here. So the chicken gamma C olfactory receptor open reading frame sequence is a mosaic of homogenized and non-homogenized regions. So how did that happen? Here's what it looks like in the pigeon. Another way to visualize this is on a graph. So on the y-axis, we have the percent similarity. And so here's a conserved region followed by a diverse region, conserved, diverse. And if you look at mammalian olfactory receptors in the human, you don't see this pattern at all. And if you just look at the third codon position, sorry, this is like super technical, but the third codon position is usually redundant and doesn't encode for, and it can encode for different amino acids, so it doesn't matter. This pattern is preserved even at the third codon position, showing that this is a phenomenon at the nucleotide level, not the amino acid level. So this pattern is actually is indicative of a different phenomenon called gene conversion, which is the non-reciprocal transfer of DNA from one homologous sequence to another. And this occurs during meiosis. So the DNA repair machinery picks up a piece of the sequence and puts it in a neighboring sequence. And this would explain that mosaic pattern. And so the hallmark of gene conversion, the thing that really supports gene conversion, is that if you take an alignment, so an alignment of all 6 species gamma C olfactory receptor repertoires, and you build phylogenetic trees from different regions of the same alignment, you get completely different trees. So here, if we take the conserved region from these 6 species and build a tree, we get the tree looks like this with these species-specific clades, very short branch lengths. But if we build a tree from just a diverse region, we see a totally different tree, different branch lengths as we would expect, but also a different topology, the species-specific clades have broken down. And one other thing about gene conversion is that it occurs within a chromosome, intrachromosomally. So so you would expect substitutions to be specific to a certain chromosome, and that's exactly what we see. For example, this G is specific to chromosome 34, C here for chromosome 16. All right, so we think gene conversion is very important in the evolution of these bird olfactory receptors. Okay, we made it through the whole technical evolutionary genetics part. Let's get to more of the fun stuff. So back to the counts. So just like in mammals, birds can also have many hundreds or over 1,000 olfactory receptors in their genomes. So pigeons consistently have very high counts of olfactory receptors, with over 1,000 in the Nicobar pigeon there, Coloanus, and over 1,000 in bandtail pigeon, Patagionis. Ducks also consistently very high olfactory receptor counts. One of the highest of all is in mallard, which of course is everywhere, with almost 1,200 olfactory receptors. And several pottards in Athia genus also have very high counts. But the new king, the new champion of olfactory receptors, blowing the elephant out of the water, if that's even possible, is the kiwi, with 3,750 olfactory receptors. And if we could just take a moment to appreciate this incredible olfactory receptor repertoire of the kiwi, where the gamma C count alone greatly exceeds the number of olfactory receptors in the elephant, and recognize that the vertebrate with the most olfactory receptors is a bird. But then at the low end of the counts, there's also birds with very few olfactory receptors. So passerines tend to have fewer olfactory receptors. So just locally, white-breasted nuthatch has very few olfactory receptors. Then the lowest counts of all, consistently in crows, with only 7 in the carrion crow there and only 10 in the jackdaw. And these low counts are also analogous to the lowest counts of all in mammals, which are in toothed whales that don't have a functional olfactory system. But the problem is everything I just showed just now is all just from the genomic repertoire. And your genome is the same no matter what cell of your body you're looking at. It's always the same, no matter what nucleus you're looking at. And olfactory receptors are expressed in different tissues and have functions outside of smell. And so this graph here, each column here is a different olfactory receptor. Each row is a different tissue, and blue is showing RNA expression of that olfactory receptor in that tissue. And you can see heart, liver, lung, kidney, whatever you want to choose, there's olfactory receptors expressed there. So we have just discovered all these brand new gamma C olfactory receptors, but how do we know if they're actually being involved in the sense of smell? So something that we can do sort of preliminarily are these phylogenetic generalized least squares methods. So one thing we just did, we know that in mammals, the number of olfactory receptors in mammals is correlated with the size of the olfactory bulb. And so the olfactory bulb is downstream of the olfactory receptors. It's used to process the information coming in from the olfactory receptors. And here we have the olfactory bulb of a black vulture. Right here is the brain and the bulb. And here we have the olfactory bulb of a turkey vulture and the brain. And we can see that the turkey vulture's olfactory receptor is much larger than the black vulture, just as we would expect. Turkey vulture relies on smell more than black vulture. And so this is what we did. We compared the olfactory receptor counts on the y-axis with the relative olfactory bulb size on the x-axis. And we see a highly positive correlation where the birds with the larger olfactory receptors, so the largest olfactory receptors of all in birds is in the kiwi, also has largest number of olfactory receptors, and the bird with the smallest olfactory bulb, the crow, has the fewest olfactory receptors. And we can also I'll also show another pattern that has been shown in mammals, where nocturnal mammal species have more olfactory receptors than diurnal. So in our phylogeny, we have 5 gains of nocturnality in the kiwi, in Caprimulgiformes, the nightjars, lateralis, black rail, Strix and Tyto, the 2 owls, and then the Strigops kakapo. And when we compare the nocturnal species with the diurnal species, we see an increase of olfactory receptors in nocturnal species. So these are good signs that the olfactory receptors are being used in smell. But what we really need to do, sorry, got a gruesome slide coming up, but we're in a room with 200 dead woodcocks, so I guess we're okay. So what we really want to know are those olfactory receptors expressed in the olfactory epithelium? And so to do this, we have to obtain bird olfactory epithelium. And so that's what this is showing. So Brian O'Shea at the North Carolina Museum of Natural Sciences really helped me here, ornithologist there. And so he gave me carcasses without data to perform this dissection to obtain the olfactory epithelium from inside the maxilla, or the top part of the beak of the bird. So here I am practicing on a killdeer and a green heron and a scoter duck. And then once I had practiced, now I was ready to obtain olfactory epithelium for study. So we got chickens from NC State, brown-headed cowbird from right here at Penn, and a zebrafinch from my campus at Duke. And we obtained the olfactory epithelium from inside the maxilla, as well as a control pectoralis muscle, and perform RNA-seq to measure the RNA in this tissue. When we do that, we consistently see that the olfactory epithelium has much higher expression of olfactory receptors compared to the pectoralis in all 3 species that we measured. And when we look at the individual olfactory receptors, here we have the genomic repertoire phylogeny, and on the outside we have the expression of each olfactory receptor, where blue is low, red is high, and black or gray is absent. And we can see that it's not just 1 or 2 or a handful of olfactory receptors expressed in the olfactory epithelium. It's like almost the entire genomic repertoire, including the alpha, gamma, and the gamma C. The unique and new gamma C is present in the olfactory epithelium. So this is good, but the olfactory epithelium is a complex place. There's anterior, posterior, middle olfactory epithelium. So different regions. And also within these different regions, there's different cell types. So the olfactory epithelium has sustentacular cells, microvillar cells, basal cells. But we want to know, are those olfactory receptors in the olfactory sensory neurons the correct cell type? And if they are, we would expect them to look like it does in this mouse image, where the air is flowing here. This is the air flowing, it was where the air would flow across. And just inside from the airflow, in this band here, are the olfactory sensory neurons. So I would like to look for olfactory receptors, but because the olfaction research in birds is so backwards, no one has even found olfactory sensory neurons in the adult bird yet. So before we could look for olfactory receptors, we had to find the olfactory sensory neurons. This took a very long time. First, we went to the anterior part of the beak, couldn't find it. Then we went to the middle, couldn't find it. All over. And then finally, when we went to the posterior part, close to the eye, we found something. So what we did here is we obtained chickens, we open up their upper part of their beak, their maxilla, and then we take the tissue out and then we cut very thin 18 nanometer slices of the tissue. And then we put that tissue on a slide and then stain it using in situ hybridization. And the probe for in situ hybridization here is CNGA2. And CNGA2 is a known positive marker of olfactory sensory neurons in the mouse. And so we use the chicken ortholog to look for the olfactory sensory neurons. And so finally, when we look at the posterior part of the beak, we found them just inside from the perimeter as we would expect. And we finally believe that we found the olfactory sensory neurons in the chicken. So now that we found the olfactory sensory neurons, now we're ready to look for the olfactory receptors. So we designed a probe against gamma C olfactory receptor. And here, this blue is the nuclear stain. So each blue dot here is a nucleus. And the green is showing our probe, as it says up here. When we do this for gamma C and look at the probe there, the pattern is nearly identical to CNGA2, showing that the bird olfactory receptors are not just expressed in the correct tissue for smell, but the correct cell type. And we also showed this for the alpha and gamma olfactory receptors as well. So that's great, but of course, what we really want to know is, do these olfactory receptors respond to odors? And if so, what odors do they respond to? So how do we do this? We have to perform the first ever functional study of bird olfactory receptors because no bird olfactory receptor have been matched to an odor before. So there's too many olfactory receptors to express. There's hundreds of different olfactory receptors. And so what we do is we do this consensus sequence approach where we take a group of olfactory receptors, in this case, all of the chicken gamma C, and then align them and then pick the most common amino acid residue at each position in the alignment. So for example, in this cartoon, here's an alignment of olfactory receptors. The most common amino acid residue at this position in the alignment is alanine, so our consensus sequence gets an alanine. Here it's a cysteine, so our consensus sequence gets a cysteine, and so on and so on and so on until we built the entire consensus olfactory receptor. We then synthesize that olfactory receptor and then ligate it, place it in a plasmid, and then transfect that plasmid into mammalian cell culture growing on a 96 well plate. 12 by 8, 96 wells. And so when the mammalian cells have transcribed, translated, and trafficked the olfactory receptor to the plasma membrane, now we got 96 wells, we can potentially put a different odor in each well and measure the response. And if the odor binds to the olfactory receptor, it will initiate a signal transduction cascade that will generate cyclic AMP. And our assay measures the amount of cyclic AMP generated. So this is what we did for the chicken gamma C olfactory receptors. So this is a blow up of just the chicken gamma C, and we made a consensus sequence of everything downstream of this node right here. And we exposed this thing to hundreds of different odors. Yeah, but let's skip that part and get to the part where the odor that actually responded, which was these group of odors called pyrazines. And so the way you read this graph is on the x-axis, we have increasing concentration of the odor from low to high. And on the y-axis, we have the response of the olfactory receptor relative to an empty vector control. And you can see that when this olfactory receptor is exposed to these pyrazines, specifically these methoxypyrazines, with increasing concentration of the odor, there's an increased response relative to the empty vector. And these pyrazines smell like green peppers, so the whole lab smelled like green peppers when we do this experiment. So then we got the response from the consensus sequence, so then we wanted a little more resolution. So we then generated consensus sequences from 4 subclades within the chicken gamma C, 1 in blue, 2 in green, 3 in red, and 4 in pink, and 4 is nested within 3. And then we exposed these to the pyrazines. And when we did that, we found the best, most robust dose response curve from that green clade 2 receptor. And you can see it increasing its response relative to the empty vector with increasing concentration for the clade 2. So this is great, but the problem is this consensus sequence, it isn't actually real. It's like it just exists in this tube in the lab and then in the FASTA file on my computer and stuff. And it's not actually in any real chicken in nature or on a farm or in a nugget or whatever. And so now we're done with consensus sequences. We're ready to hone in on that clade 2 and we created primers designed specifically to those clade 2 olfactory receptor sequences. And now we're done ordering from IDT, done synthesizing. We're amplifying these native olfactory receptors directly from chicken genomic DNA. And so we cloned dozens of these and tested them, and we finally got a response from a handful of them shown here. So in the previous graphs, the different colors on the graphs were different olfactory receptors. Now the colors on the graphs are different odors. So we're looking at 3 different methoxypyrazines. And we found responses from these 4 native chicken olfactory receptors to these pyrazines. And although they're all responding to the pyrazines, they're responding a little bit differently. So OR1 and OR4 are most sensitive to that pink pyrazine, whereas OR2 and OR3 are most sensitive to the blue pyrazine, showing that there's diversity in binding properties within the chicken gamma C olfactory receptors. And this is the first ever match of a bird olfactory receptor to an odor, and would suggest that the chicken is capable of perceiving what we perceive as the green pepper smell. But this is good, but the problem with this is like a Frankenstein kind of experiment. So the bird olfactory receptor is in the mammal cell. And so is there a way for us to bring the pyrazine result back to the live chicken? And so the way we did this is we performed PS6 in the chicken. So what we do is we take a chicken and we put it in a box for an hour. And this is an airtight box. And so the chicken, whatever it was smelling before it went in the box, it got habituated. And then after 1 hour, there is a treatment. And so either the chicken gets no odor, or it gets acetophenone, which is a control odor commonly used in mouse smell experiments, or it gets exposed to the pyrazine. And so after that hour stimulation, we then sacrifice the chicken and then perform PS6 on the olfactory epithelium. And the way PS6 works is this is a ribosome, and this is a small ribosomal subunit. And attached to the small ribosomal subunit is a protein called RPS6. And when neurons are activated, the RPS6 protein is phosphorylated. And so if we target phosphorylated RPS6, we can label activated neurons. Okay, and that's what the chicken looks like when it's in the box. So that's, there you go, for a visual. And this was the result we got. So on the left-hand side, we have the same thing as the in situ hybridization images, just a different magnification. We were able to find the gamma C olfactory receptors again. And then in the middle, we have the PS6, which shows no activation or very limited activation in the no odor control, just background activation, which is difficult to see even though the lights are off, in the acetophenone control odor. But then with the pyrazine, we see quite a bit of activation of olfactory sensory neurons. And importantly, this activation overlaps with the distribution of olfactory sensory neurons expressing gamma C olfactory receptors, showing that olfactory sensory neurons that express gamma C olfactory receptors activate when following exposure to pyrazines in the live chicken. So that's great for the gamma C olfactory receptors. But if you remember, the gamma C olfactory receptors are only in birds, but that is not the case for the other 2 groups, the alpha and gamma receptors. And the literature for they're shared amongst mammals, reptiles, fish, amphibians, whatever. And the mammal olfactory receptor research is like light years ahead of the bird olfactory receptor research. We're like in the Stone Age. And so we can leverage this information from mammals to learn about alpha and gamma olfactory receptors in birds. So one great example is this human olfactory receptor OR51E2. This is like a model olfactory receptor. We know like everything about this olfactory receptor. So we obviously know the sequence. We know the exact odors it binds to. We have the crystal structure of this olfactory receptor. So we know exactly how the orientation is right here of the odor in the binding pocket and which amino acid residues interact with which parts of the odor. So we know a lot about this olfactory receptor. And so what I did was I took the chicken olfactory receptor repertoire that we've been looking at the whole talk, and to this phylogeny, to this alignment, I added the human OR51E2. And when you do that, the human OR51E2 on the chicken phylogeny, it appears here in red. So it doesn't appear here. It doesn't appear here. It appears here, with a clear one-to-one orthologous relationship with one single chicken olfactory receptor, which I call chicken OR51E2. And so then the question arises, mammals have OR51E2. It responds to short-chain fatty acids, propionate and acetate. Chicken has OR51E2. Does the chicken OR51E2 respond to the same odors across 320 million years of independent vertebrate evolution? And so when we do this, we were able to recapitulate the known human olfactory receptor response with increasing concentration relative to the empty vector. We see a response to acetate and propionate. When we test the chicken olfactory receptor OR51E2, we see the same response. We see the response to the same odors, showing that the odor binding properties of this olfactory receptor are preserved across hundreds of millions of years of evolution. So in the future, I would like to perform odor exposure and behavioral tests on the chicken because we don't know why the chicken needs to perceive this pyrazine odor. What is the chicken doing for this green pepper smell? So future experiments will address those questions. And then other experiments will, I would like to take these methods that I've used in the chicken and apply them to diverse species across the phylogeny, so like the vultures and the honey guide indicators and albatross. And then I just want to thank my lab, Hiroaki Matsunami lab, my PhD advisor, Chris Balakrishan, the whole lab, especially Mona Marie. She did a lot of that in situ hybridization work, and my funding and collaborators and all that. And I could take any question. Okay.
Monkey, he smell green pepper, yet they don't have a gamma.
Yes. So it would be a non-canonical or a novel way of detecting the pyrazine smell that is not shared with, yeah.
Maybe some biologies in some of these regions, sub-regions.
Well, that's the active area, yes, that we want to pursue.
Okay.
Right. So not necessarily the whole sequence, but the residues specific to the binding pocket may be the same or they may be different. Either way, it's interesting because either way, it's a novel way of detecting a novel binding pocket shape, or it's the same. Either way would be interesting.
I mean, another question. When you showed the different species, race of different species, there are a lot of intra-species evidence of some type of recombination conversion.
Yes, so we're
Can you repeat the question?
Oh yeah, I'm sorry for the Zoom people. Yep. So I forgot the question. Oh yeah, yeah, we can smell green peppers, so so we must be using a different mechanism for detecting the green peppers in the chickens because we don't have gamma C. So that was the first question, and then this question. Oh, did I just forget it?
Species.
Right. Within a species. Yes, so we were not able to answer this question until just a few months ago. And so now we're trying to answer it because the long read genomes available were only one per species. But just in the last few months, Scott Edwards' lab at Harvard has uploaded like 100 different scrub jay genomes, all from either Florida or Western scrub jay. And so we're going to go into those scrub jay genomes and find that out for the first time. So check back. Yeah.
That was amazing, everything you've done. So there's a million questions, but I'm gonna Let's ask one. It probably isn't that hard. So the gamma Cs are unique to birds,
Yeah.
but so in the beginning, I think you were talking about looking through the genome to compare to receptors in other species.
Yeah.
How would you compare and find gamma Cs if they're unique to birds?
Right. So they still have many regions that are shared. So that even though they don't come out in the phylogeny, when you look at the entire sequence, there are certain motifs that are preserved across all olfactory receptors. And the query file that we used had sequences that were similar enough to recover them. Yes. And actually, we took sequences from the literature, and zebrafinch was already in the literature, so zebrafinch was. So that's another thing.
Well, I spoke about genes. Are there also a lot of genes?
Yeah, there's a lot of. Yes, so a lot of them have been pseudogenized, and I was analyzing those, but those files became so huge because there's so many pseudogenes that I also don't have a satisfying answer yet for that question. So they are not counted. So they may be counted because it's complicated. So my filtering mechanism will filter out any uh, five or more insertion or deletion within a transmembrane domain, and that's how I characterize a pseudogene, or one of the ways, or several ways. But like there are some critical amino acid residues, the ones that are preserved across all olfactory receptors. And if you have a mutation in just that position, your whole olfactory receptor could be pseudogenized. So my pipeline doesn't filter for that. So there's a little bit of contamination of pseudogenes in the counts, but you can only do so much with the refinement. Yeah. Yeah.
Let's see if I can ask this question correctly, because this is like really wild research. But okay, so the olfactory receptors of, say, crows are very low, and then you were trying to show, whether the olfactory receptors correlate to sense of smell. Are there any birds that don't smell?
Well, I would.
Sense of smell, I should say.
I don't know, and it's very hard to figure that out. So also something that I should mention, which is kind of a caveat of the talk, is that a lower number of olfactory receptors doesn't necessarily mean a worse sense of smell.
So that's actually what I was wondering.
So the indicators, like the honey guides, they have actually not that many olfactory receptors, like below average, but we suspect that maybe they only really need to detect wax. They only really need to be tuned to a few different compounds. So they may be very sensitive to wax, but don't have a lot of olfactory receptors. And then it's kind of unclear, like, what does a good sense of smell even mean? Does it mean you can detect a lot of things? Does it mean you can detect a few things really, really well? So, yeah, it's kind of hard to tell. I don't know of any birds that can't smell. And because that's like the thing I'm trying to fight against, I won't say that there are.
And then you already asked the corollary question that I had, which is there a relationship between the number and the amount of smell?
Yeah, it's unclear. I think you could probably safely say that a carrion crow with just 7 olfactory receptors is not good at smelling as a kiwi with 3,750, but it's not exactly clear what the relationship is. Yeah.
So we just talked about the number of olfactory receptors and how well a bird can smell, which is obviously inconclusive. But do we have any explanation for why there may be more olfactory receptors in certain families versus others? And if you could start your answer with the question was.
Oh my gosh, I'm sorry, Zoom people. Forget it. I can't remember what just happened. I'll repeat Kevin's. So yeah, okay. Okay, so there isn't a clear correlation between the number of olfactory receptors and sense of smell. So why do certain lineages have lots of olfactory receptors, and is there some other signal that determines that? Roughly.
Yeah.
Okay.
Ding ding ding, you got it.
Okay. Right. So we don't know why the kiwi needs 3,700. We don't know why they need to have that many. But there clearly is some phylogenetic signal. because the pigeons consistently have high counts. The other ratites, which are fairly divergent from the kiwi, but emu, cassowary, rhea, ostrich, we looked at all those. They all have a high number of olfactory receptors, nowhere near 3,000. But there's some phylogenetic signal there that shows that the common ancestor would have had a lot of olfactory receptors. So there is some phylogeny aspect to it. But I have looked at, and nocturnal, I had the slide that shows that nocturnal can increase it. But I've looked at all sorts of different ecological variables, different diets, different habitats, different range sizes. Is it migratory or not? What's the latitude that it's found at? Altitude, eye size. I looked at all these different things. And I can't find a correlation with number of olfactory receptors in those things. So either something's wrong with my analysis or, yeah, jury's still out. Yeah.
Can I read a question from Zoom?
Sure.
If you don't mind, Phil Whitmer asks, so which comes first, the behavior or the olfactory receptors? Crows eat a lot of different foods. Is that because they can't smell that well?
Uh, so I was really surprised by the crow result. I thought the crow was going to have a lot of olfactory receptors because it like loves to eat trash and all sorts of things. So it was a surprise. So I don't know why that's the case with the crow. I know this isn't really answering the question, but one thing is there seems to be a trade-off with olfactory receptor counts and intelligence. The idea is that the olfactory bulb and olfactory system is expensive to manage, and that if your brain is doing all sorts of other things, it invests elsewhere. And so the crows have low counts, but parrots also have low counts, another highly intelligent lineage. And then we have low counts compared to other mammals. And the whales have low counts. Actually, the baleen whales also have some low counts too, in addition to whales. And so yeah. So
is there a correlation in that regard to receptors versus overall brain complexity?
Is there correlation in that regard to olfactory receptors in terms of brain complexity?
Maybe their brain is better at taking fewer signals and figuring out what they mean.
Yeah, I don't know if that study's been done, but if they did it and they showed it, I would believe it. And then the other question was, which came first, the sense of smell or the olfactory receptors, right? Wasn't the first part of the crow question?
Behavioral.
Oh, the behavior or the olfactory receptors. I think it's well. This is just this is a common question in evolution. Which came first? Exactly. I don't know. Yes. I think the behavior starts. You have some rudimentary level of olfactory receptors, and then as the behavior becomes more of a selective advantage, you get more and more specialized tuning to fine tune. Sorry, I just said tuning, fine tune that behavior.
I was going to say, does it correlate with specificity of their food supply? But that doesn't seem to maintain for the eaters or wax.
Yeah. Yeah.
And you have the visual aspect too, in combination with the olfactory aspect. I mean, we swear that the fish crows at the shore know the Wawa wrappers and go after
by vision. Then recognize that logo. Maybe geese do too. Hey, there we go. Yeah.
Question on Zoom.
Yeah.
Carol, you want to unmute and ask a question?
Hi. I'm going to ask a question that veers into really layman's perspective on things territory just a bit, but it has to do with the fact that, and even people, when we think about olfactory things, we think of smell, we think of things only that we're perceiving through inhaling something through our nose and sensing chemicals that are in the air. But in fact, a lot of what we think of as taste is actually olfactory, rather that we only taste a very few things. And I think about this because I read the book called An Immense World by Ed Young that talks about animal senses a lot. And for instance, things like snakes and elephants have very high recognition of things that they touch, like the elephant touches something with its trunk and puts it in its mouth, or the snake touches things with its tongue, but what it's analyzing is analyzing by olfactory um, um, means. So I was wondering whether some of the distinction between the birds that have more receptors or fewer might have something to do with whether they are doing more with their tongues or beaks rather than what they're sensing from the air. For instance, kiwis pecking in the ground. Uh, that this is a very vague question, but do you have any thoughts about that?
I think it's possible. I would check out Maud Baldwin's work. She is the bird taste expert and has shown really amazing things. Like, for example, sorry, this isn't directly answering your question initially, but like birds. So birds, just real quickly, because this research is so amazing. Birds come from theropod dinosaurs, and this was a carnivorous lineage of dinosaurs. When you have a carnivorous lineage, you often lose the ability to detect sweet sugars. So, for example, cats can't detect sweet. So birds have lost the ability to detect sweet. So then Maud Baldwin's question was, well, then how does the hummingbird be attracted to the sweet flowers? And they repurposed their taste receptor that usually detects umami to be able to detect sweet in a novel way of detecting sweet. So she's the bird taste expert. She has a bunch of taste papers like that. But that would be something that probably no one has looked at all. So you're talking about a correlation between olfactory receptor count and the amount that a bird species uses its tongue while feeding. Would that be an accurate
Yeah, or
distillation of?
how much that it needs to know is coming by what it touches with its tongue as opposed to what it's breathing in, right? So is it locating things by flying around, or is it something that's pecking in the ground and locating things by what it, by what it's touching with? I mean, ducks probably would be another example of something that is touching the mud on the bottom of and discerning what's good and what isn't, um, with it more with tongue than by breathing in. So yeah, that was what I was wondering. So
Yeah, so I'm not sure. I think it might be tough to find a correlation there because like foraging is my favorite thing to think about bird smelling, but the bird smell can be used for all sorts of things. And so you may get a lot of olfactory receptors because say migration or something like that. There's been incredible papers showing that birds use olfaction to migrate. And so I know that's not a good answer to your question. I don't know if there's an answer. I think it's a great thought. I think it's testable, but I can't tell you whether or not it's an actual relationship. I'm sorry, I haven't looked at it. Say.
No worries. So this might not be a fair question because it probably relates to research that is complementary but not your project. But if you're challenging sort of the sensory hierarchy, right, saying that olfactory senses is actually more important than bird processing information generally, is there any research to suggest that the long-standing assumption is incorrect on the other side, that birds actually don't see as well as perhaps we assume they do?
No. So like I'm kind of oh my gosh, thank you. So I was saying that people said before that birds use vision at the expense of olfaction, but am I trying to say that maybe with an increased level of olfaction, there's some expense to vision?
Or is there any research to suggest that they actually that there's a better balance
I see.
of vision to olfactory,
Right.
rejecting sort of the assumption that they saw well and smelled poorly?
Yes. So I I don't really like the trade-off idea. I'm fine just saying, hey, they can smell, they have this sense of smell, their vision is still just as good as it is. I think that so maybe my presentation was a little bit unfair to say that vision is still extremely critical to birds. It is the main sense for many different behaviors. I'm not trying to take vision down at all. It's just that smell is also important. But there are some examples, like the kiwi has these tiny little eyes, very poor vision, yet has this amazing olfaction. So clearly there's some trade-off there, but I don't mean to diminish vision at all.
Last question.
About more about migration.
Okay. All right. Last question. Okay, so check this out. So there's this experiment where they took catbirds from Illinois and brought them to New Jersey, and then they released the catbirds during migration. So the catbirds going to migrate to Central America. And so when they took them from Illinois and put them in New Jersey, they found that the catbirds compensated for that translocation. So whereas they would go directly south, they were in Illinois, they then shifted to have a westward trajectory when they were brought east to New Jersey. But then they took some of these catbirds and they put a zinc solution in their nares and this makes it so they can't smell for a period of time. And they found that those catbirds did not change their orientation after translocating. They just went directly south. And this research built off of research in pigeons, which showed that if you disrupt the olfactory nerve or do this zinc thing, they can't navigate to their roosting site. They need olfaction to navigate. But in both of these studies, when they disabled the magnetic detection in the birds, there was no change. So the studies highly argue against a magnetic mechanism for migration in birds, like strongly. They believe that that's mostly a myth. And they also found out that when the pigeons could navigate back to their roost, but they couldn't get all the way there. If you don't show the pigeons, if you blind the pigeons or cover them and don't let them see and then translocate them, they can get close to the roost, but they can't get there. They need visual cues when they get close. So they use olfaction as they come into the roost, and then when they're local, they use vision landmarks to find the roost.
So are vagrant birds are birds that have a bad sense of smell?
Could be, yeah. Yeah, let me get their genomes. Let's find out. So
Like what?
they didn't know, so I emailed that researcher. I was like, "Let's find out what your pigeons are smelling. Let's find out what the cues are." And she was like, "I'm retired." But I don't know. Yeah. Yes.
So with seabirds like albatross, they leave and they're separate after nesting, and then they come back.
Yeah.
Is it possible that they can recognize each other then by smell and not just by sight?
Yeah, so they are thought to navigate to their breeding islands through smell. So these same researchers in the pigeon, they also removed, did the zinc thing with shear waters and found that the shear waters were just wandering around the ocean. They couldn't find their way back. And if you've been to one of these like little nest site islands, I mean, there's just poop everywhere, right? The nests are all these burrows all over the island. And so that island has a distinct smell. And so it's thought that they use olfaction to not only find the island, but this has been shown in leech's storm petrels that once they're at the island, because leech's storm petrel is mostly nocturnal and breeding. So once they get to the island, they use olfaction to find their particular burrow in the dark.